Hello everyone, let's get down to business to defeat genes, environment, and development. Alright, so the chapter preview. And evolution in species heredity. Species heredity refers to the genetic endowment that members of a species have in common, including genes that influence biological maturation and aging processes. Species heredity supports the theory that some aspects of development and aging, as certain patterns of development and aging, are universal. Humans don't fly. So basically the idea here is that there are some species-specific universal developmental trajectories within a particular species. So there is a 0% probability that your child is randomly going to sprout wings and then fly away. This is not X-Men. Darwin's theory. Darwin argues that there is genetic variation in a species. Without this, an environmental challenge could wipe out an entire species. Some genes aid adaptation more than others. Some are good, some are bad. Genes that aid their bearers in adapting to their environment will be passed to future generations more frequently than genes that do not, through a process known as natural selection. So basically the idea here is that genetic variation exists and has to exist within a species, or a particular stressor, a particular trauma, a particular environmental challenge could single-handedly wipe out an entire population if the entire population was 100% genetically consistent. Evolution and species heredity. The environment and genes affect evolution. Evolutionary psychology is the application of evolutionary theory to understanding human thinking and behavior. One limitation is that it's biased towards individual interpretations. They argue that brain flexibility / adaptation thought to be the legacy of human evolution. So our key takeaways here are this field known as evolutionary psychology, which interprets or tries to interpret things based on kind of their understanding of evolutionary principles. But there are obviously biases in that, because you're going to look at individuals' behaviors through the lens of whatever your social, political, economic, ideological views, and then interpret those individual behaviors within a prism of your kind of ideology that you're bringing to the table. Because you're kind of speculating on stuff that happened millions of years ago, hundreds of thousands of years ago, kind of within a prism of your contemporary theory. So you'll often see, certain gender roles or statuses within society, whether it's racial hierarchy, caste, economic differences, justified in kind of evolutionary terms, but obviously it's difficult to prove and often not falsifiable, because we cannot go back in time. We don't have a time machine yet to go back and test the reason for specific evolutionary patterns and how exactly those evolutionary patterns are useful or are representative in the 21st century. Individual heredity. The genetic code. Sperm cell and an ovum each contribute 23 chromosomes to the zygote, leading to 46 total chromosomes. The sperm and ovum are produced by meiosis. Chromosomes are slender threads inside the cell's nucleus that carry genes and the genetic material. The DNA is a double helix-shaped molecule, so kind of just some basic definitions here. Continuing on with basic definitions, cell replication. Mitosis, a cell divides to produce two identical cells, each containing the same 46 chromosomes. Zygotes become multiple cells through mitosis, so kind of straightforward. We're reproducing ourselves on a cellular level. Interestingly, the Human Genome Project was this large-scale project, and a link is in the description, where researchers map a sequence of chemical units / letters that make up strands of DNA and a full set of 46 human chromosomes. But what are some of their findings? 3% of the human genome consists of what was defined as genes, parts of the DNA that are transferred into RNA, acts as a template for protein production. The other 97% played critical roles in regulating activity, originally called junk DNA. So we have kind of 3% of what is kind of typically referred to as the genes, and the rest kind of playing a supporting role. 99.9% of the genome is the same across all humans. So let's look at two types of twins, identical and fraternal. One easy way to say this is MZ and DZ, or monozygotic, or dizygotic. See, I went for it anyway, even though I just recommended using the little abbreviations. But identical twins, they originate from one zygote. One fertilized ovum divides to form identical individuals, and it's 1 out of 250 births. Fraternal twins, two ovum are released at approximately the same time, and each is fertilized by a different sperm. 1 out of 125 births. So kind of just the difference here is that one set of twins originate from one zygote, while another set of twins are fertilized by two different sperms because there's two different ovas. Let's look at sex chromosomes. Chromosomes of the 23rd pair are the sex chromosomes. Male X chromosome and Y chromosomes. Females have 2 X chromosomes. And the father determines the gender of the child because the sperm carries an X or Y chromosomes. So again, kind of just basic biology here. Genotype versus phenotype. Genotype is our genetic makeup. The phenotype are our observable traits. Gene expression is the activation or deactivation of genes by environmental experiences throughout development. So genes are influenced, kind of like our influence, or influence, I should say, and are influenced by kind of biochemical environments all around them. All cells have the same gene, which genes are expressed decide which of the cells make your blood cells, neurons, etc. And I'm going to get into gene expression towards the end of this lecture. Single gene pair inheritance. Thousands of human characteristics are influenced by only one pair of genes, one from the mother, one from the father. There's a dominant gene and recessive gene, kind of basic bio stuff here. Dominant genes express when paired with a recessive gene. Recessive genes are a weaker gene that can be dominated. However, many or most psychological traits, in contrast, probably aren't influenced simply by dominant recessive genes, influenced by hundreds or thousands of genes. So I worked in a gene environment lab as a PhD student. And this is kind of a common understanding. For complex psychological phenomenons or concepts, there is no one X gene that causes whatever behavior. Rather, genes are implicated, and I should say networks of genes, complex networks. You can almost think of them as complex brain networks being associated with certain phenotypes. Now, what is causing those phenotypes, what is the directionality, is debated. But on the right here, you can see some tables of kind of just the basic biology of dominant recessive traits. Polygenic traits. Polygenic traits are influenced by multiple pairs of genes interacting with multiple environmental factors. Examples, height and weight. So I just grabbed, I'm ecocentric, as Piaget would say. And I grabbed my thesis here that looked at the impact of a military occupation in the Democratic Republic of the Congo on mothers who were pregnant during that occupation, which is a military occupation, and it occurred for basically about 2 weeks. And you'll even see in this chapter that things like height are referred to as more genetically based. And what they mean by genetically based is that it's less influenced by environmental outcomes. But my researchers found that in this case, there was an association between mothers experiencing that military occupation and giving birth to children who were shorter, so reducing their height. So it's kind of the environment interacting with genetic expressions of height or prenatal development, which we'll get into again in future slides and chapters. Mutation, a change in the structure or arrangement of one or more genes, can be beneficial depending on the environment and type of mutation. Odds that mutations will occur are increased by radiation and toxic waste. Most are just spontaneous errors during cell division. Odds of mutations increase as parents get older. Typically a different version of a particular gene. So all we're getting at here is that there are mutations that at the genetic level that is most likely to occur due to environmental pollutants. The bottom photo is animals in land around Chernobyl, and those animals are understood to kind of have different genomes than other members of their species who have not interacted with that nuclear radiation in the environments surrounding the Chernobyl power plant. And most mutations, however, are just kind of different versions of a particular gene. So it's kind of like natural, kind of random variations in the gene, and if the mutation works out, typically, most of the time they don't, the cells will just die out, or the organism itself will die out, and the mutation will not be passed down genetically. Copy number variations. Instances in which parts of the genome is either deleted or duplicated, differs from mutations, can extend over a large stretch of DNA, can either be inherited or rise spontaneously, increases risk of polygenic disorders, Alzheimer's, bipolar disorder, and schizophrenia. So what we're getting at here is that this is kind of can be thought of for the purpose of this class, kind of a more extensive, larger mutation. It's extended over larger patches of the DNA, and instead of acting on like one gene, like a mutation would, kind of generally speaking, this can occur across wide swaths of DNA, which then can impact a number of genes, which can then increase the risk of disorders that involve multiple genes. That's the polygenic disorders, such as Alzheimer's, bipolar disorder, and schizophrenia. Now let's jump into chromosomal abnormalities. Result when a child receives too few, too many, or abnormal chromosomes at conception. Examples are Down syndrome, Turner syndrome, and XYY syndrome, so you can literally see it within the name of the syndrome, and is the main cause of pregnancy loss. So genetic diseases in testing, genetic counseling, which provides information about the nature, the likelihood, effects, and treatment of genetically based diseases and disorders. Carriers do not have the disease, but can transmit the gene for it to their children. So this kind of begs an ethical question. To what extent should genetic testing be used to identify disorders? And so should we test for it, and what should we do for that information? It begs begins an ethical debate, and especially if we eventually have the ability to modify certain genes, if that's even possible, with these complex genetic networks that we barely understand. But it begs the question: what do you do about that knowledge? How far can genetic testing go, and how far should it go? So moving on to sickle cell trait versus disease. So kind of alluding to what I said before, carriers. Sickle cell trait is found in humans close to infestations of mosquitoes, so West Africa, the Mediterranean, Middle East, East Asia. It protects against partial protection, or it provides partial protection against a type of malaria. Environmental insults such as low oxygen environments, their red blood cells deform into a sickled shape and become adhesive, sabotaging the cell's ability to carry sufficient oxygen and causing them to block small blood vessels, including capillaries. These events trigger excruciating painful episodes. So basically the point here is that you can have sickle cell trait, which is adaptive, against certain types of malaria. But in a different environment, that sickle cell trait can be maladaptive. That is in environments where there are low oxygens, or there is low oxygen, mountain, hiking, whatever. So that's kind of just an example of a genetic adaptation being adaptive in a certain environment but not in another, and the impact of the environment on the expression of a gene from a trait to a disease. Prenatal detection of abnormalities. So we're going to just look at three methods: just ultrasound, which is sound waves to scan the womb; maternal blood sampling, which analyzes DNA of offspring from placenta; and pre-implantation genetic diagnosis, vitro fertilization in lab, test cells which form from mitosis. And you can actually kind of do a lot of interesting experiments with that we'll talk about in future classes. But just for example, you can implant the sperm of a fertilized egg from a mom who, let's say, she was smoking, or she was a smoker growing up, or she even smoked during pregnancy. So say she's carrying a zygote that was fertilized within the lab from two non-smokers. Well, if you put that non-smoking zygote, that is a zygote from two non-smokers, and you put it into the womb, injected, I should say, into the womb of a mother who is smoking, you see the negative that baby or that fetus, I should say, at birth has signs. And it has similar, actually really kind of identical negative consequences from being exposed to smoking in the womb as would a fertilized zygote who was from two parents who were smoking. So the idea here is that the prenatal environment matters. So even though that sperm and egg, that fertilized egg, is from two parents who previously had never smoked, say they never smoked in their life, but if you put that fertilized egg into the womb of a mother who is smoking, that prenatal environment still negatively impacts that fertilized zygote from the two non-smoking parents. So the idea here is that the prenatal environment matters. Behavioral genetics, the scientific study of the extent to which genetic environmental differences among people are responsible for differences. We have heritability, which is the percentage of the variability in a trait across individuals that is due to genes. Some traits are highly heritable, height, I already alluded to that in previous slides, but sure, while others are not, religious affiliation. Height among plants may be largely heritable. Watering these plants and environmental manipulation can result in substantial increases in their heights. High heritability doesn't imply unchangeability. So basically what we're looking at here is just literally the percentage. We're trying to calculate a percentage of a particular behavioral outcome that is caused by genes or influenced by genes, and what percentage is influenced by environment. Very difficult to do, if not impossible. But kind of the key takeaway here are that high heritability doesn't imply unchangeability, and even in traits that are highly heritable, such as height, and kind of the example of watering a plant, if you do not water your plants, as we've seen in quarantine, they will die and they will grow less tall. So what's one way that we measure, or what's one method that behavioral geneticists use to measure heritability? One way is through twin studies, which determine whether identical twins reared together are more similar to each other than fraternal twins. That's because identical twins share 100% of the same DNA, while fraternal twins don't. So then you can kind of calculate the difference between, say, Self-esteem in identical twins is 90% the same on whatever, some self-esteem tasks. Well, fraternal twins, I'll just make up something, 30% the same. So then you do a little calculation, and you're like, okay, and again, it's broadly stated. Okay, so therefore, genes contribute to 60% of self-esteem development. That's kind of the idea here. The limitations. Identical twins could be more psychologically similar because they shared prenatal environments. Some scholars argue that prenatal environment is the most important predictor of future developmental outcomes. That's generally referred to as the fetal programming hypothesis or the developmental origins of disease hypothesis, which we'll cover more in future classes, or chapters, I should say. It does not account for the environmental impact, intergenerational life history, or genetic expression, or the genome. So all this means is that not only is the prenatal environment important, but studies have found that intergenerational stress is passed down intergenerationally and is passed down at a genetic level. There's plenty of studies that have looked into this, and we'll cover some of those in this class. But most famous ones are one trauma called well, I should say, just keeping it broad. In populations who starved, or mass starvations in some populations, there are still genetic markers found of this mass starvation generations later within individuals. So is it that, okay, so if you were to just look at the mother, say 3 generations removed from that starvation, you might conclude, okay, that mom really didn't have any starvation within her immediate environment, but you're not accounting for the environment from generations ago. So how you disentangle that, very difficult, and might be impossible. Identical twins are often treated similarly. Heritability technically says little or nothing about how malleable, alterable a trait is. So again, heritability does not mean something is unchangeable. And heritability is not a fixed number. If a range of environmental influences on a trait within a population are reduced, the heritability of that trait will increase because most of the genetic differences in that trait will be due to genetic factors. So you can imagine one bucket full of, hypothetically environmental factors, and we'll just say another bucket full of heritability factors. So if you reduce the range of environmental influences that you're measuring, that bucket will be smaller, an environmental bucket, correct? Which will automatically fill up the bucket of heritability because it's kind of just this ratio. It's 50% environment, or whatever. We're starting at a baseline of 50% environment, 50% heritability. And by removing the environmental influences that you're measuring, that trait will automatically be thought of as being due to genetic factors. That doesn't mean like this methodology is like garbage or whatever, but this is just kind of a limitations inherent in trying to study heritability. Another is adoption studies. Are children adopted early in life psychologically similar to their biological parents or their adopted parents? So you can find groups of identical twins, then say they're adopted in either the same household or different households. So one twin goes to a location in Bolivia, and another child goes to Antarctica. Then you can measure their self-esteem, see how correlated it is, and then calculate based on that how correlated they are. So say how similar they are. So say they're 90% the same. Then you can do some calculation, boom, boom, boom. Then you have a heritability score of self-esteem that it would not be influenced by similar environments because they're raised in literally completely different countries. Limitations. Genes in the prenatal environment of the biological mother could influence development. So back to that developmental origins of disease theory of development. Adoption agencies tried to place children in homes similar to those they were adopted from. So typically, the adoption agency recognizes the background of the children who are being adopted, and they don't want to throw them into a just completely random household where they have no familiarity, they've never heard the language, the culture is completely different. That would make it very challenging for the child. So although they're in a new different home, they're still likely to be maybe in a similar religious household, a similar income level, a similar country, etc. Children are typically adopted to above average environments, which could underestimate the full effects of environments. And because disproportionately children are adopted by parents who are wealthier or higher classes, those children would be receiving kind of just like optimal environmental influences, environmental inputs. Thus, it could be underestimating the effects of the environment, because in environments where there are less inputs, so like imagine being adopted into, I don't know how this would happen, but imagine you're adopted into a family who is homeless. Well, you'd probably see worse outcomes for the child across the board, and that's just because you're lacking critical resources, critical interactions, critical environmental factors which we know shape development. So because of that, children getting adopted into wealthier households, higher class households, you're kind of reducing the range at which the environment factors are the range in which environmental factors are present or not present. So, say you have 100%, so you're going to be adopted into a home with, I'm just making up something here, 100% of the environmental factors you need to optimally develop. But at the lowest, you would be adopted into a home that has 90% of the environmental factors you need to optimally develop. So what about environments that are 80%, 70, 60, 50%, offer 0% of the environmental inputs? So because of that restricted range, we might actually be underestimating the extent to which the environment is involved in the development of whatever specific psychological outcomes. So family studies. They include pairs of siblings who have a variety of different degrees of genetic similarity. You can follow them longitudinally. Measure qualities of family siblings' experience to determine similarities and differences in sibling environments. So kind of the idea here is you go into a family home, you follow them longitudinally, and you are able to assess, did they go to after-school programs? How does the father interact with one child versus another child? How does the mother interact? Are there family dynamics? So it's just following them longitudinally at a family level, which allows you to collect more data and, again, trying to determine how much the environment is implicated in these developmental outcomes, or genes are implicated in these developmental outcomes. All right, so let's go by that. So we're going to look at temperament here, just introducing it. We're going to get into this much later in class. But temperament is tendencies to respond in predictable ways, such as sociability and emotional reactivity, that serve as the building blocks of later personality. Cross-cultural effects, again, we'll get into that in later chapters. This is kind of just giving a little framework here of future chapters. Findings indicates that living in the same home does not make children more similar in personality. So there's evidence that some researchers argue that because children will, some studies have found that children differ on personality tests that suggest that the environment, the home, is not impacting the development of personality. Psychological disorders, just looking at one here. Genes and environment contribute to psychological disorders. For example, schizophrenia. Genes contribute substantially. Average concordance rates for identical twins is 48%. Environment plays a role. So even in identical twins, for something that we know that genes, in a disorder that we know that genes contribute to, the concordance rate is only 48% of twins. So aka environment plays a role. And these statistics, percentage, are always questionable. And in the book it says you can't actually measure the heritability. There is no exact percentage. It's impossible, but in the beginning of the chapter, but then they proceed to give us a number of percentages. So, just take the percentages slightly with a grain of salt. and recognize that the book is contradicting itself immediately throughout this chapter. Gene-environment interactions. Gene-environment interaction is the effects of genes depend on what kind of environments we experience. How we respond to the environment depends on what genes we have. Often it's a combination of high-risk genes and high-risk environments to trigger psychological problems. And what I'm going to emphasize throughout this course is that it's the interactions among predispositions of genetic risk factors being activated and by high-risk environments. So literally stressors in the environment interacting with genetic predispositions being implicated in behavior. And now let's get into some more specific examples and to see what I mean by that. Okay, so we have genotype/environment correlations, which process by which genetic factors contribute to variations in the environment. People with different genes have different experiences. So let's look into some specific, two different types here. But passive and evocative, starting with passive, occurs when children passively inherit the genes and the environments their family provides. Because parents provide children with both their genes and a home environment compatible with those genes, the home environments to which children are exposed are correlated with and are typically likely to reinforce their genotype. So all this means is that, again, imagine that your parents are contributing to 50% of your genes, each one of your parents contributing 50% of your genes, but they also are contributing to 100% of the design of your house, which theoretically are going to reflect, their genes and, their life history, etc. So the idea here is not only are they giving you those genes, they're giving you at least your immediate environment, which theoretically reflects their life history and genes. So because of that, a developing child is passively receiving these environmental influences around them, namely their home environments, their rooms, etc., all designed by their parents, who also each have contributed to 50% of their genes. So you can see the correlation here between passively receiving these environmental influences. It's passive because the child is not active in it. The child is laying there, they're crying, whatever. But they're not designing, they're not going to IKEA and picking out the merchandise here. Compared to evocative genotype environment correlations, which is how the social environment reacts to individuals based on their inherited characteristics. Whether one has a more outgoing or shy temperament will affect how he or she is treated by others. So for this correlation, this is your environment reacting to you. So you see maybe a child is outgoing, and then you are outgoing with the child. If you see the child is shy, then maybe you're more chill with the child, or you approach it. You approach the child more slowly, and not loud or whatever. So it's just kind of the environment reacting to you. It's not passive. It's more of an active. It's not active because we're about to hit active genotype, but it's the environment is acting on you based on your personality type or your inherited characteristics, I should say. Now we're going to get to active genotype. Active occurs when individuals seek out environments that support their genetic tendencies. The individual with a genetic predisposition to be extroverted is likely to go to every party inside, invite friends over, etc. That's highly socially stimulating. So what we're talking about here is that for active genotype is that this is an individual acting on the environment. So we had evocative, where the environment is acting on us. We had or responding to us, kind of interacting with us. We have passive, where it's kind of just like washing over us. Your house, your pillows are not interacting with you, but you're kind of receiving the design of those pillows, the design of your house, etc., are kind of interacting with you and reinforcing your genetic predispositions. Here, it's an individual acting on their environment. So say, let's go back to the example of a child who's talkative, but let's instead call it extroverted. An extroverted individual is more likely to go to parties, or more likely to interact with people, and barring the people at the party not hating them and not being obnoxious, they'll have a good time, and then they'll feel great about it, and then they'll be more extroverted. So it's an individual acting on the environment, selecting environments that emphasize their genetic traits, and then having a good time. So getting that feedback from the environment, which reinforces that theoretical genetic predisposition, and then it also strengthens that social tendency. So it's round and round we go. Genotype environment interactions. It involves genetic susceptibility to the environment. The concepts of gene environment interaction tells us that people with different genes react differently to the experiences they have. By contrast, the concept of gene environment correlation says that people with different genes have different experiences. Adoption studies provide evidence for genotype environment interaction. So what are we getting at here? The point is that for genotype environment interactions, which is typically just called gene environment interactions, nature and nurture combine. They interact, for our statistics people out there. For example, a stress triggers depression only if a person has a high-risk genetic predisposition for depression. So that in that case, this interaction, this gene-environment interaction, is occurring because an individual has a predisposition for depression. So they can receive the same exact stressor. However, it's going to cause some individuals to become depressed and some individuals not to become depressed. One reason for that is because certain individuals are predisposed genetically, this concept argues, for depression. So it's literally a gene an environment kind of activating that gene, and we're about to get into that, but that's a good framework, activating that genetic expression. So you're kind of at risk, but then that environment makes that risk factor become a reality. It turns it on, and now you have depression. It's your genetic predispositions in your environment interacting. In contrast, the gene-environment correlation is about people's experiences, as I just stated. And kind of the same example that I already gave. Children who have genes associated with extroversion are probably going to have extroverted parents. They're going to seek out extroverted experiences. They're going to go to the bars. They're going to receive responses from other individuals because of their extroverted characteristics. So it's kind of more nature affecting nurture. Predispositions, your genetic predispositions influencing your experiences. Okay, epigenetics effects on gene expression. And just kind of bringing this up right now, and we're going to talk about it throughout this course. Epigenetics, the process through which nature and nurture co-act to bring forth particular developmental outcomes. Epigenetic effects, environmental factors affect whether or not particular genes are expressed. Might explain differences on identical twins. So again, it's this idea, the way that I typically teach this is that epigenetics can be seen as a light switch, the environment turning something on or off. Even though you're predisposed genetically for depression does not mean an individual is destined to be depressed. However, if they run into environments that are challenging, that are stressful, that are toxic, such as poverty, they experience abuse, they experience high levels of stress, that accumulation of stress activates an individual's genetic predispositions, in this case for depression, and thus they're depressed. So it's a risk factor being activated, being turned on like a light switch by environmental influences. And you see this at the genetic level. In research that I've conducted, or labs that I've been part of in the Democratic Republic of Congo, have found my colleagues have found that exposure to war in the Democratic Republic of the Congo impacting mothers and children at a genetic level, that genetic expression, environments, I should say, leading to the expression of genes, turning on those and shifting a genetic risk factor, a genetic predisposition from a disposition, from a risk to being on, to from going from being at risk for depression to being depressed. Okay, so that's all for this week. Thanks for tuning in, and I'll see you next week. Remember to hit that like and subscribe, or smash it, whatever they say. All right. Anyway, I'll see you next week. Bye.